Hello, the audience. We are here to present, even though we cannot be there physically, but it's our privilege to come in the virtual way and then present our project to you. I'm Shannon San, and
I'm Gene McGinnis, also Michael McGinnis. Ni hao.
I'm Derek Hilton.
yeah, 大家好, and we are from Spelman College in Atlanta, Georgia, in the United States. Today we are presenting the article, not the article, but the project preliminary findings entitled Contextualizing the Learning of Circuits Under Biological System. We apply the Yenka software for student-centered modeling practice and self-assessment. We will be doing more of the hands-on and then demonstrate what's going on with the students when they are playing around with Yenka. And Derek will show you a little bit of the basic usage for Yenka. Jean will be showing you how this is applied, the physical setting into the biological system. Before we even begin this project, we had the interdisciplinary reasoning and communication in mind because we hope that the project can facilitate students' knowledge integration, the integrations in which that they can integrate knowledge from one discipline to another. In this case, is the biology and physics integration. And they can also translate the terms that are used in the biological system and the physics system. They can also transfer what's going on in the neuron or the circuit in order to understand the action potential that's being found in the neuron. And but ultimately, that we expect every student can have the cognitive process to transform or change the context conceptually or even physically. So the physical transformation using the Yenka software is what we want and expect students to do. The purpose of this project was to introduce a computer-supported modeling and by means of Yenka, and the biology majors are our target here in their freshman year. And so ultimately, they can apply their understanding of the neurons with the integration of the circuits. This project investigates students' perceptions toward practicing the interdisciplinary modeling, and we are using the computer technology interface, so the students will be able to authentically collect data and then assess their understanding for the action potential, membrane potential, and equilibrium potential. Then we are coming to the context. So we want to situate every of the audience what this project is about. This project, we recruited 164 students from freshman year, and they are all the biology majors taking their second semester biology. There were three electrical models that were used to understand equilibrium potential, membrane potential, and action potential. But in this presentation, we only show you the basic resting membrane potential and action potential in the neurons. Derek will show you the basic circuit, while Jean will show you the membrane potential and action potential.
Okay, when you first open the software, we get a blank screen like this, and to create a circuit, we can click Object to get a toolbox, and here's our toolbox. So we can have many components that we can build our circuit with. Okay, so your choice. So let's start out with a symbolic circuit. So we have our list of things that we can build a circuit with, basic things right here. So let's start with a battery, 9-volt battery. We can start with a switch and just drag it over here to build the circuit. And then we can have a bulb here. Now we have our components. The next thing to do is to connect them. So we can just click and create wires to connect these things. Click, create a wire. Now that our circuit is built, we can flip the switch and the bulb gets lit. Open the switch, bulb gets off. We can also build the same circuit using pictorial symbols. We can have our 9-volt battery. We can create a switch, also a bulb. We connect them, and now when we turn the switch on, the bulb is lit. Open the switch, the bulb is off. In addition to building circuits, we can make measurements, and we can have an ammeter that we can drag over here and put in the circuit. And there we go. So when we turn the switch on, the ammeter now reads 90 milliamperes. We can also have a voltmeter that you can measure the voltage across the bulb. Okay. So let's move it up here a little bit. We can also have measurements using graph. So we can say measure the current in the circuit. Say right here. You can drag it. So now we have to change the scale. So we can double click on the scale from 0 to about, say,
Negative.
So we need negative 500. That's too much. Minimum to be, say, negative 100. And 0. There we go. So when we open the switch, we're back to 0. And close the switch, we're down to. We're measuring the current in the wrong place. If we had measured the current over here, we'd have got a positive number. So that's the basic introduction to circuitry.
Okay, so we want our first-year biology majors to understand membrane potentials, action potentials, other things going on in nerve cells. And what we've done is pre-made a Yenka circuit for them to explore. And here we've got a picture of a neuron with an axon. And this is where the electrical signal produced by nerve cells travels to other nerve cells. Then this is zoomed in on the membrane of that cell. And you can see we have potassium channels and sodium channels, with sodium ions more concentrated on the outside, potassium ions more concentrated on the inside. So potassium ions would like to diffuse out, and that would make the inside of the cell more negative. Sodium ions would want to move in, which, since they're positively charged, also would make the inside more positive. So the students can change these what are called equilibrium potentials. So by sliding this bar, you can change this equilibrium potential, which basically reflects how large the ion concentration is across the membrane. So for sodium, it's around it's about plus 70 millivolts. And for potassium, it's a minus 80. It's probably too small for you to see, but these batteries are reversed. So this really is a negative 80. These little resistors in here represent the conductance of those channels. And conductance, of course, is the inverse of resistance. So the higher the resistor, then the lower the conductance. And the students are able to change those. So like now there's a very low resistance, which means there's a high conductance to potassium. And we can change that. And then sodium, we can also shift that sodium resistance, which is down here. So we can increase it or decrease it. And as we do that, the resting membrane potential, which is shown up here, will change. As you can see, as I'm changing these resistances or conductances. We can change the membrane potential to about anything we want. Now, at rest in a regular cell, usually we have a lot of conductance for potassium and very little conductance for sodium. And so we end up with about a -60, -70 membrane potential. Specifically potential that we call the action potential, and that's what travels along the nerve as a signal. So in this diagram, we've reversed the side. So potassium's on this side now, sodium's on this side. And you notice there's extra channels. Here's the same channels we had before. We call those leakage channels for sodium and potassium. That sodium's always leaking in, potassium's always leaking out. But we've also added these voltage-gated sodium channels and these voltage-gated potassium channels. These are channels that open or close depending on the voltage in the membrane. And these are found only in nerve cells and muscle cells. So we've put in these little switches to simulate the opening and the closing of those channels for either potassium or sodium. We've also wired up, like Derek showed, a graph down here which shows the membrane potential. So at rest, it's about -80. So if I take it off pause, then we see the voltage. Now, if I add some sodium channels, then that's going to make sodium more dominant, and the voltage, you can see, went up towards the sodium's equilibrium potential. If we close that, it goes back down. Now, if we add these extra potassium channels, the voltage goes down just a little bit. So we give the students the job of trying to choreograph the opening and closing of these channels so that it simulates what would be found in a real nerve. So this is the action potential, time in millivolts. We, of course, don't use times of milliseconds. Our students aren't quite that fast. So this time base is in seconds. But basically, in a real membrane action potential, it's at rest, then the voltage increases dramatically because the sodium channel is opening. It drops because sodium channels close. Then potassium channels open and it goes actually below the resting membrane potential, and then it drifts back up. So we asked the students to simulate that, and since my fingers aren't as fast as the students, I stop and pause. So here is the membrane potential at rest. We're going to open these sodium channels, open the conductance. So then you can see the membrane potential goes up very quickly. Then those sodium channels change state. Now they're closed again, which means the resistance is open. And these potassium channels now close. Now, resistance is closed, the channels are open. And then we drop back to low level, and then eventually these things again reclose, and we drift back to rest. So in a sort of a crude way, this shape is reproduced over here. And this just helps the students understand the role of the channels opening and closing to change the membrane potential. Now, what we hope is that the students, with this little introduction to the biology of the circuit, are also picking up some information about voltages and resistors and currents and such. So then later, when they take physics in a year or so, they will be able to use this same circuit in the physics department. They'll use this, basically the same circuit, to help them understand circuit theory and Kirchhoff's laws, and throw in some capacitances so we don't have the stair step. And they'll take it further, but hopefully they'll have a context for their for the circuit, because they'll understand not just light bulbs on and off, but they'll understand the biology of it. So here's a circuit like they may use in physics, where they actually physically build it. So these would be equivalent to the equilibrium potentials, these two voltage sources. here's some resistors, here's some switches which cause the channels to open and close. So they'll be moving from sort of the simulation in their first year to building more advanced circuits in their physics course in their third year.
In which that they are filling in the high permeability, which means that the conductance is high and then the resistance low. So they are able to do the authentic assessment in which that we can detect students are understanding it fully or not to analyze the data. Students' perceptions are also one of the main components we are interested in. So we use the five-level Likert scale ranging from extremely helpful and not helpful at all for the question such as, what do you think about the Yenka model's neuron activity in the lab? And the students are answering that on the survey. The results show that in the beginning, the student had the lower performance on the 10 questions, and then there's significant difference after they engage in the Yenka activity. Based on the response for the 5 Likert scale survey, the students, at least 76% of them consider the Yenka to be very somewhat helpful for their understanding of the neurons. And so this study that showed the power of the computer-assisted technology by means of the Yenka. So it's a simulation software that helps us to teach and model the physics and biological phenomena in class. It also supplements the conventional instruction that's usually taught linearly without the integration of other disciplinary knowledge. So this supplements the conventional instruction of neurons by means of the circuits with a simulation context. So we orient the students in the not only the context of neuron, but we introduce the the physics disciplines knowledge, so they can integrate. And they can also translate the knowledge, translate the terms such as the resistance or the concentration, all of that in applying that to the construction of the action potential or membrane potential. They are also empowered by the graphic to interpret what the graph means. So this is also another authentic way for student assessment. For the future study, as Jean pointed out, that this was a preliminary study that's administered in the freshman year. Now they are moving to their junior year, and they will collect more data, concentrating more in the physics context to see how much the students can retain their knowledge. And also we elaborate by means of the case studies or the interviews. We hope that we can elaborate this framework with the integration, how the translation works, and the transfer of their knowledge. And ultimately, hopefully, students will be able to change whatever they retain the knowledge in order to apply to the different contexts in their real life by means of the technology-enhanced interface. So the student-centered assessment is crucial in this project for us to understand how the students can model the biological context by means of the physics experiments. And since we cannot be there physically, we still hope to hear from you. That's why we enlist all of our emails. If you have any questions for Derek or for Jean, please email us and we'll be happy to address any of your questions. And you can also download Yenka online for a 30-day trial version, but we use this as a site certificate that we purchased. And the acknowledgement for this.
Yes, we'd like to acknowledge the NSF grant and also the participants, Rosalind Bass, Michael Bruns-Karin. These are people who help us create the activities. Paul Camp, who helped us with the pedagogy, and the instructors in the biology labs, Elita Tillman, Anna Powalny, Mentowa Bayulu, and Alicia Griffin. And we acknowledge them. Thank you.
Thank you so much. 再见。
再见。
再见。 见。
再见。
再